Hello everyone, welcome to 10-Minute Physiology. In today's video, we're going to be talking about how bicarbonate is handled in the nephron. So with that, let's give it a go. So I would like to begin by first looking at the proximal tubule. So how does the proximal tubule deal with bicarbonate? So in the basolateral membrane, we have the sodium-potassium pump. The sodium-potassium pump moves 3 sodiums out of the cell into the interstitial fluid, and then it moves 2 potassiums into the cell. And it does this by using the energy released by ATP hydrolysis. So the sodium-potassium pump establishes an electrochemical gradient for sodium, and this electrochemical gradient is utilized by the sodium-hydrogen exchanger. So the sodium-hydrogen exchanger brings in 1 sodium, and by doing this, it releases energy. So when sodium is brought down its electrochemical gradient, this releases energy. And this energy is used by the exchanger to transport 1 proton into the tubule lumen against its electrochemical gradient. So once the proton or hydronium ion is in the tubule lumen, it will combine with bicarbonate to form carbonic acid. Now the carbonic acid will be broken down into water and carbon dioxide through the action of carbonic anhydrase. So the carbon dioxide and water now have to get into the cell. The carbon dioxide gets into the cell through 2 ways. The first way is by passively diffusing through the plasma membrane into the cytosol. And the second way is going to be through aquaporin proteins. So it's through these 2 ways that carbon dioxide gets into the cell. Now water will get into the cell through the action of the aquaporin protein. The aquaporin protein allows water to move down its channel into the cell. Now, once water and carbon dioxide are in the cell, the water and carbon dioxide can react together in a specific reaction forming carbonic acid. And this reaction is catalyzed by carbonic anhydrase. So the carbonic acid will then break apart into hydronium ions and bicarbonate. The hydronium ions can be recycled back into the tubule lumen through the action of the sodium-hydrogen exchanger, or they can go through the proton pump and be pumped out into the tubule lumen by using the energy released by ATP hydrolysis. So the bicarbonate will then move into the interstitial fluid primarily through the sodium bicarbonate cotransporter. Now the sodium bicarbonate cotransporter co-transports 3 bicarbonates and 1 sodium into the interstitial fluid. So it keeps this ratio here. So that is basically how the proximal tubule is going to reabsorb bicarbonate. So please note that the proximal tubule is going to be responsible for reabsorbing most of the filtered bicarbonate. The proximal tubule reabsorbs around 80% of filtered bicarbonate. So now let's move on to the thick ascending limb of Henle. So how does the thick ascending limb of Henle deal with bicarbonate? So the thick ascending limb of Henle is going to be very similar to the proximal tubule in how it deals with bicarbonate. So on the basolateral membrane, we have sodium-potassium pump. The sodium-potassium pump moves 3 sodiums out of the cell and 2 potassiums into the cell by using the energy released by ATP hydrolysis. So the sodium-potassium pump establishes an electrochemical gradient for sodium, and this is going to be used by the sodium-hydrogen exchanger. The sodium-hydrogen exchanger exchanges 1 sodium for 1 hydronium ion. So the hydronium ion moves in the tubule lumen, and once it's in the tubule lumen, it can combine with bicarbonate. And when they combine, they form carbonic acid. Now, carbonic acid can be converted into carbon dioxide and water through the action of carbonic anhydrase. So the carbon dioxide can then move into the cell through passive diffusion, and once carbon dioxide is in the cell, carbon dioxide can react with the water that is present inside the cytosol, and it can form carbonic acid, and this reaction is going to be catalyzed by carbonic anhydrase. So carbonic acid will then dissociate into hydronium ions and bicarbonate, and the hydronium ions can be recycled back into the tubule lumen through the action of the sodium-hydrogen exchanger and through the proton pump, which uses the energy released by ATP hydrolysis to do this. So it's through these 2 ways in which hydronium ions can be pumped and transported back into the tubule lumen. Now the bicarbonate can move into the interstitial fluid through 2 main mechanisms. The first mechanism is going to be through the anion exchanger 2. So anion exchanger 2 exchanges 1 bicarbonate for 1 chloride ion. And the other mechanism is going to be through the sodium bicarbonate cotransporter, which co-transports one bicarbonate and one sodium into the interstitial fluid. So this is the primary ways in which how bicarbonate is going to be reabsorbed in the thick ascending limb of Henle. So when all is said and done, around 10% of filtered bicarbonate is reabsorbed by the thick ascending limb of Henle. So now let's look at how the early distal convoluted tubule is going to handle bicarbonate. So on the basolateral membrane, we have the sodium-potassium pump. The sodium-potassium pump moves 3 sodiums into the interstitial fluid and then 2 potassiums into the cytosol. And it does so by using the energy released by ATP hydrolysis. Now on the apical membrane, we have the sodium-hydrogen exchanger, which exchanges 1 sodium for 1 hydronium. The hydronium then can combine with bicarbonate to form carbonic acid. Carbonic acid is broken up into carbon dioxide and water, and this reaction is catalyzed by carbonic anhydrase. Then carbon dioxide will move into the cell through passive diffusion and go into the cytosol. Now, once the carbon dioxide is in the cytosol, it can combine with water and form carbonic acid, and this reaction is catalyzed by carbonic anhydrase. Carbonic acid then can dissociate into hydronium and bicarbonate. The hydronium can then be recycled back into the tubule lumen through the sodium-hydrogen exchanger. Now, on the basolateral membrane, we have the anion exchanger. The anion exchanger will exchange 1 bicarbonate for 1 chloride. So that's how bicarbonate is going to be reabsorbed in the early distal convoluted tubule. So now let's take a look at the late distal convoluted tubule. So in the late distal convoluted tubule, we have the sodium-potassium pump that is present on the basolateral membrane. And on the apical membrane, we still have the sodium-hydrogen exchanger, which exchanges 1 sodium for 1 hydronium. The hydronium combines with bicarbonate, forming carbonic acid, and then carbonic acid is dissociated into carbon dioxide and water, and this reaction is catalyzed by carbonic anhydrase. And the carbon dioxide will then move into the cytosol through passive diffusion. Once this carbon dioxide is in the cytosol, it reacts with water to form carbonic acid, and this reaction is catalyzed by carbonic anhydrase. The carbonic acid will then break down into hydronium and bicarbonate, and the hydronium can be recycled back into the tubule lumen through the sodium-hydrogen exchanger, and it can be recycled back into the lumen through the proton pump. Now the bicarbonate can move into the interstitial fluid through the anion exchanger. The anion exchanger exchanges 1 bicarbonate for 1 chloride. So this is the mechanism in which how bicarbonate is going to be reabsorbed in the late distal convoluted tube. So around 6% of filtered bicarbonate is reabsorbed by the distal convoluted tubule. So now we're going to see how the cortical collecting duct handles bicarbonate. And we're going to start off by first of all looking at the alpha cells that are present in the cortical collecting duct. So how do the alpha cells deal with bicarbonate? Well, on the apical membrane, we have the proton pump. The proton pump pumps out protons into the tubule lumen by hydrolyzing ATP and using that energy to pump out the protons. So the protons will also be pumped into the tubule lumen through the action of the proton-potassium pump. So the proton-potassium pump basically exchanges 1 proton for 1 potassium, and it does so through the action of hydrolyzing ATP and using that energy to do this exchange. So this pump basically pumps out 1 proton, and then it pumps in 1 potassium into the cell. Now on the basolateral membrane, we have the anion exchanger. The anion exchanger is going to exchange 1 chloride for 1 bicarbonate. So the anion exchanger basically takes 1 bicarbonate from the cytosol and moves it into the interstitial fluid, and then it brings 1 chloride into the cytosol from the interstitial fluid. In addition, we also have the sodium-hydrogen exchanger, which basically takes 1 proton and moves it out of the cell into the interstitial fluid, and then it moves 1 sodium into the cytosol. And also on the basolateral membrane, we have NKCC1. NKCC1 co-transports 1 sodium, 1 potassium, and 2 chlorides into the cytosol. So what we see from what we learned here is that the alpha cells are going to be secreting acid into the tubule lumen, and they're going to be reabsorbing bicarbonate and bringing it into the interstitial fluid. So alpha cells secrete acid into the tubule lumen. So now let's look at the beta cells that are present in the cortical collecting duct. So how do the beta cells deal with bicarbonate? Well, on the apical membrane, we have the pendrin protein. So the pendrin is basically going to exchange one bicarbonate for one chloride. So the pendrin moves one bicarbonate into the tubule lumen, and then it moves one chloride into the cytosol. In addition, we also have NDCBE. So NDCBE basically exchanges one bicarbonate for one chloride and one sodium. So the bicarbonate moves into the cell, the chloride moves out of the cell, and the sodium moves into the cell. So this is one way in which bicarbonate is reabsorbed by the beta cells. So on the basolateral membrane, we have the proton pump, which pumps protons into the interstitial fluid by using the energy released from ATP hydrolysis. In addition, we also have the sodium hydrogen exchanger. The sodium hydrogen exchanger is going to exchange one proton for one sodium. It moves one proton into the interstitial fluid and one sodium into the cytosol. In addition, we also have the NKCCC protein, which basically co-transports one sodium, one potassium, and two chloride into the cell. So what we see here for these beta cells is that these beta cells sort of do the opposite as the alpha cells. Where the alpha cells secreted protons into the tubule lumen, the beta cells do the opposite. The beta cells are going to secrete bicarbonate into the tubule lumen, and they're going to basically reabsorb protons, moving them into the interstitial fluid. So now we're going to look at the final region for today, and this is going to be the outer medullary collecting duct. So in these cells of the outer medullary collecting duct, we see the proton pump on the apical membrane. The proton pump moves protons into the tubule lumen, and it does this by using the energy released by ATP hydrolysis. In addition, we also have the potassium pump. The potassium pump basically pumps in one potassium and pumps out one proton, and it does so by using the energy released by ATP hydrolysis. In addition, we also have bicarbonate in the tubule lumen. The bicarbonate is going to combine with the proton, and it's going to form carbonic acid. Carbonic acid can then be broken down into carbon dioxide and water through the action of carbonic anhydrase. And then the carbon dioxide can move into the cell through passive diffusion, where it will combine with water and form carbonic acid. So the carbonic acid can then be dissociated into protons and bicarbonate. Now the protons are going to be recycled through this pump here and the proton pump, where they can be pumped back out into the tubule lumen, whereas the bicarbonate is going to be exchanged by anion exchangers for chloride. So these anion exchangers move bicarbonate into the interstitial fluid, and then they bring in chloride. So when it's all said and done, around 4% of filtered bicarbonate is reabsorbed by the outer medullary collecting duct. So that's it for this video. I hope it helped you understand how bicarbonate is going to be handled along the length of the nephron, and I hope to see you in the next video. Thank you for watching, and I hope to see you next time.